about the gastrulation, okay? Although we wrote the topic gastrulation long ago, but we had gastrulation now exactly. But that was all preparation, what is going to happen, right? So if you remember, what we have now is I'm just going to change the direction, like the way you have drawn it, I'm just going to flip it upside down, right? So what we have here is, right here is so top here, then all of this should like this, the outer, this is your trophoblast cell, although there are no more trophoblasts, they're kind of like embedded there and everything is formed, I'm just drawing them like this. And then what we have here is, right here is your okay, so these are your epiblast cells. So I'm just, making that upside down what you have. And then you have your visceral endoderm. So this is your visceral endoderm and your parietal endoderm. Okay. Now, so this is your now bilaminar germinal disc, epiblast, and visceral endoderm. Now, epiblast cells, they are going to give embryo. So embryo will come from epiblast cell. Now what happens, now think about you are looking this thing from the top, okay? So where you are, you are kind of like sitting here and looking it from the top. If you look it from the top, this will look like this surface, okay? Because it will be like this, many cells there, kind of like this, right? So what happened, so this is the midline, right? So this is, for example, the caudal end of this. So what happened, what you see, you're observing it, and what you see during almost the day 15, okay? So almost day 15 this thing started, 14 and 15. What you'll observe, you will observe kind of like, if you have a piece of paper and you kind of scratch it, you find a streak formation there, right? So if you have a piece of paper, you scratch it very hard, right? You press it very hard, right? You can see a streak there. So what happened, a streak form, that streak form from the midline, start from here, and that streak goes like this, okay? And that streak has a special kind of shape. The shape is like this. This is like this. The streak is formed like this. So this streak is formed, and this streak is called primitive streak. A primitive streak is formed. Okay? So start from the midline and go toward this side to the bilaminar germinal disc. Now, this streak is interesting because this streak actually, right now you get what side will be the anterior side, what side will be the posterior side. That means what side will be the cranial side, what side will be the caudal side. Okay? So, this streak formation takes place, and if I draw this streak like this thing like this, for example like this, okay, so this will be the right side, the future right side. And this will be the left side. Okay? And this side, on this side right here, this side is going to give you the cranial end. And this side will give you the caudal end. And remember, I told you that if you have a mutation in dynein when you're talking with the sperm motility, I told you when the mutation in dynein is there people have chances that 50% time their heart will be on the right side, right? And here I'm saying you, the formation of this streak is telling you left and right side. So that means something happens right here, right? And we'll talk what happens, right? So that the heart is always on the left side, not on the right side. So this thing happened then. And then what happened following this, so this is just a streak, right? And then what happened, the same thing is there. So the place right here, so for example, if you look at your fingers, it's exactly like this, right? You will see here, kind of like more depression, here less. So same way here the depression start happening. So here, the depression means that cells from here, they start moving in. And they form a depression. So cells have started moving in. That's what the gastrulation is, right? And this thing, this circular depression is formed. Circular depression. And this depression is called primitive pit. Okay? Now, this depression starts from here, and then it moves from here to here. So all those cells are starting moving, and ultimately this thing is formed. This all cells are now moving. Now it's exactly like your fingers. Like here the gap is there, it's like that. The things are moving from here like this. And that is called primitive groove. Okay? Now cells from here, they are moving from drawing inside, cells are moving here, cells are moving here, cells are moving here, cells are moving here. So cells are going down under. Okay? So it means that the cells which are moving on this side, on this area, they are going to specify the head area, the cranial area. And the cells which are moving on this side, they are going to specify the caudal area. So now if we look from this position, what is happening there, right? This was up, what is happening there. This is exactly what happens in the bird. In the bird, the primitive streak is formed like this, and the cell moves like this. So that's why we are kind of like making kind of copying what is happening in the birds, right? So although they have this thing happened because if you have seen the egg of the bird, so you will see a big yolk there. You might have, if you have used organic, free egg, whatever there, sometime you will see right on the yolk here, you will see a black spot. And that black spot is actually the embryo. And that is right on the top. Right here is all yolk. This is the only area where that is, and that kind of like, if you're looking here, it actually starts developing from like this, from top. Because there is yolk. But we don't have any yolk, but we still do the same thing. And that's why we do the hatching also, right? So this is okay. Then what happens? What is happening here? Okay? So I'm just drawing this. So this layer So this is your epiblast, right? And beneath this, you have your visceral endoderm. This is your visceral endoderm. So now what happens? I'll draw and I'll give you a little bit. So some of the cells from here, from the epiblast, they start, as we said, what is happening? These cells are moving in, right? So what happened? The some cell, now, these cells are attached to each other because there are some cell adhesion molecules, right? And those some cell adhesion molecules are called EP cadherin. So right here, these cell adhesion molecules, there is EP cadherin there. So some cells start losing EP cadherin. And definitely that start from here, from the circular depression of the, or what we call primitive pit, that start from this area, right from the center. And these cells, they start losing EP cadherin, and when they lose EP cadherin, they move down. So they lose EP cadherin from here. So EP cadherin is gone, EP cadherin is gone, and these cells, they start moving down. Okay? So when they are moving down, what is happening? We are saying this is the gastrulation has actually started. Okay? And this process, therefore, invagination. They start moving down. So what happened? So I'll now draw it again with a little bit gap here. So this is like this. So, for example, these cells had lost the EP cadherin. Now these cells have now come here. Okay? But what you had visceral endoderm there. Okay? So these were the visceral endoderm. So these cells kind of start getting into the visceral endoderm layers. Okay? So what happened? So some cells are coming down, they are inserting in visceral endoderm layer. And slowly, now these cells are also dividing, right? They are not just a fixed number of cells, they are also dividing when they're moving down. And they are also dividing in the epiblast layer. So slowly what happened, these cells which were moving down, they replace completely the visceral endoderm. So visceral endoderm is now completely replaced by the cells which were moving down from here to here. Okay? So now, what is happening? So now, right here, all these cells are now replaced. Visceral endoderm is now replaced. Now, visceral endoderm is replaced by the cells coming from epiblast. Now, when these cells are in this layer here, what they do, they actually start changing into the endoderm. Okay? And when they replace it, what they form? They form what we call definitive endoderm. Definitive endoderm. So now these cells are called, when they completely replace them, you call them as definitive endoderm. Now at this stage, when these cells have become definitive endoderm, these cells are no more called epiblast cells. Now you call these cells as ectoderm. Making sense? Now, same process. These cells still keep on moving, and then a third central layer is formed. These cells will keep on moving. As these cells are moving, they're coming in contact with endoderm. Remember, endoderm is telling the cell above, ectoderm cell about to become mesoderm, right? So they are moving, further moving, and in this way, a mesoderm is formed. Making sense? So what we are saying here is, so this is happening if you are looking from the up also. So what is happening? These cells, they are moving down, spreading this side. Now this is from a mouse. This is called Hansen node in mice, but not in the mammals. Okay, so that's why it's written Hansen node. So what is happening? So these epiblast cells come here. They have replaced these visceral endoderm and they form their definitive endoderm. So these hypoblast cells are being replaced, right? And then this whole layer will be formed endoderm. They'll move more in the center. They are going to form the mesoderm. Making sense? So let's watch a small movie here.
Bilaminar embryonic disc, consisting of the hypoblast and epiblast, has formed. Throughout the third week of development, this bilaminar disc differentiates to establish 3 primary germ layers in a process known as gastrulation. Around 15 days after fertilization, a thickened structure forms along the midline in the epiblast near the caudal end of the bilaminar embryonic disc. This is called the primitive streak. At this stage, the formation of the primitive streak defines the major body axes of the embryo, including the cranial end towards the head and caudal ends towards the tail, as well as the left and right sides of the embryo. At the cranial end of the embryonic disc, the primitive streak expands to create a primitive node, which contains a circular depression, known as a primitive pit. This depression continues along the midline of the epiblast towards the caudal end of the streak, forming a primitive groove. Once formed, cells of the epiblast migrate inwards towards the streak, detach from the epiblast, and slip beneath it into the interior of the embryo. This process is known as invagination. The first cells to invaginate through the primitive groove invade the hypoblast and displace its cells. The hypoblast cells are eventually completely replaced by a new proximal cell layer, which is referred to as the definitive endoderm. By day 16, the majority of the hypoblast has been replaced. The remaining cells of the epiblast are now referred to as the ectoderm and forms the most exterior distal layer. Some of the invaginated epiblast cells remain in the space between the ectoderm and newly formed definitive endoderm. These cells form a germ layer known as the mesoderm. Once the formation of the definitive endoderm and mesoderm are complete, epiblast cells no longer migrate towards the primitive streak. Throughout gastrulation, the ectoderm continues to form from the cranial to the caudal end of the embryo, establishing 3 distinct primary germ layers throughout the whole embryonic disc. The gastrulation process is final with the
Making sense? Question?
So the yellow cells, so like the third image, the yellow cells on the very bottom, that's gonna be a part of the embryo, right?
Three distinct primary
Okay, now
germ
This
layers
layer,
throughout the
that's a
whole
part of the embryo?
embryonic
Oh, yeah.
Okay.
This is actually the whole circle thing. They're kind of like showing it, you know? But it does not look like this. Let me show you how it looks like this. So this is kind of happening in a small disc area. Remember, you have a sac above, amniotic sac, and you have a yolk sac down. It looks exactly like a cup. This is actually, it look like. This is kind of like all the allantois membrane, amnion, and all those things. This is what is the embryo is. Right? So this, all the things, whatever is happening, they're not the part of the developing embryo. Okay? So here is interesting thing. So you see this is kind of like this area is that. So this was your node. When I was drawing it, I was saying this was the node, right? So from here, the cells were moving on this side, and the cells were moving on this side. This is exactly that. Okay? Now, again, 3 layer, endoderm, mesoderm, and ectoderm. Now, this area right here, this is, again, you look here, called precaudal mesoderm, visceral endoderm. So this is kind of an area like we talk about. We said notochord is there. We said precaudal plate is there. They're all sending signal up. So here in this area, this AVE, what we call anterior visceral endoderm. This area, AVE, because this area is the cranial area. Remember we said this will be all cranial, head area, right? So this area, the cranial area, that means what should be there in the cranial area? The cranial area should be devoid of BMP, Wnt, those things should not be there, right? And FGFs. In the mammal, the new term FGF is there. Now, what is making their inhibitor? If these things are there, if BMP is there, Wnt is there, FGF is there, you are not going to form the formation of head and all those things, remember? So it means those should be inhibited. So their inhibitors, there we had the new hoop center, that was endoderm, which was telling mesoderm to make all those inhibitors, right? So here, this anterior visceral endoderm is the signaling center. Responsible for inhibitors of BMPs, Wnt's, and FGFs. Okay, exactly same thing, but orientation is different. Making sense? So then, so we know that the anterior, posterior, that's what we are talking about, posterior axis. Right? So anterior is the anterior visceral endoderm is the signaling center that sends the signal to inhibit BIMs, BMPs, Wnt, and FGA in this area. So this area can become the cranium area, right? Now after the cranium, what we have. So if you look at the body structure, then you have this spinal cord, right? But this spinal cord is where? spinal cord is in those ring-like bones. Right? Those ring-like bones are there. And you call those bones as vertebrae. So you have those vertebrae there, right? And in mammals, even you talk about giraffe, I don't know. So how you spell the giraffe? I don't know. Or mouse. Okay. Either you have mammals, whatever, giraffe or mouse. If you look at them, each will have 7 cervical vertebrae. 13 thoracic vertebrae. Those are going to give you ribs. And each of those will have 6 lumbar vertebrae. 6 lumbar vertebrae, which are going to give all the abdomen area, right? And then you have the 4 sacral, which are pelvic area. And then you have different kind of like tail vertebrae, depending on what organism you're talking about, right? We don't have any tail vertebrae. Now, this is the tail. Now think about this. Giraffe also have 7, 13, 6, 4, and tail is whatever it can be. Now think about that. Giraffe has a very long neck compared to human or mouse. They still have 7 cervical vertebrae, so vertebral size is big, right? So from here to here, when we're talking about anterior posterior, head to the beginning of this, or maybe the spinal cord formation, we are saying that is the game of the BMPs, right? You inhibit BMPs, you inhibit wind. BMP, wind together, you get head. You only BMP, you get the spinal cord. But then how these vertebrae, which are covering them, they are specified because they are the part of that anterior posterior polarity, right? So how that happened? And we are saying that is because of what we call Hawkes code. So Hawkes code play an important role in anterior posterior axis pattern. So I'm using term here, the pattern. So pattern, what is pattern? Pattern means the pattern of vertebrae, right? Because that is determining, we are not kind of like just a one single head-like thing. We have all those, our body is divided in head, thorax, and abdomen, right? And that head, thorax, abdomen division is based on the type of vertebrae. Because your thoracic vertebrae are going to give you ribs. And your cervical vertebrae are going to give you neck. So how that is established? And these vertebrae, they are not the nervous system, that is mesoderm. Okay? We'll talk how it formed if we have enough time. But mesoderm is giving you vertebrae. Remember, if this ectoderm right here is going to become the brain and nervous system, the mesoderm right here, which is beneath this, that is going to encircle those spinal cord, right? And in the next topic we'll talk about, because here this brain is outside, it should go in, right? Our brain is not hanging out. How that happened we'll talk. But now just think that brain is formed, spinal cord is formed, and that spinal cord is surrounded by the vertebrae, and vertebrae are giving the pattern of our body, right? And how that pattern is established, we are saying that pattern is established based on the Hawkes code. So what is Hawkes code? Okay, so we are saying that all the known vertebrates means the animal which have that vertebral column, right? All the known vertebrates, they have 4 sets of Hox genes expressed per haploid set on separate chromosomes. Their location and order of expression is remarkably similar in all vertebrates. So in all the vertebrates, we have 4 sets of Hox genes. They are expressed on different haploid chromosome, and how they are arranged on the chromosome and how they are expressed in the body, that is exactly same in all the vertebrates. Okay. Now you will say, what are those? So we are saying in human, we have Hox A, Hox B, Hox C, and Hox D. These 4 different gene types are there. Now, if we look here, let me show you this thing. This is Drosophila, this is mouse. The pattern, they also have these Hox gene, we call them home family gene there. Home A, B, B. When they look at this pattern is exactly similar what is happening in the mouse or the human. So you have Hox A, Hox B, C, D. 4 different set of genes. And there are 13 different type of. You can have Hox A from 1 to 13. Hox B 1 to 13. So there are 13 genes in each set of the group. For example, Hox A is one set. In that one set, you have 13 Hox A1, Hox A2, Hox B, Hox 3, Hox 4, like that, up to 13, right? And if you look at the gene level, for example, Hox A, Hox A1, A2, A3, A4, A5, these are exactly like there. A6, A7, A9, A10, 11, 13. Hox B, B1, B2, B3, B4, 9. Hox C, C5, C6, so these are present there. Now their expression, how they are expressed, that changes the expression of the vertebrae. Now let me explain that. Look here. Chick and mouse. So you have expression of this. Let's talk about the cervical vertebrae, right? Now in cervical vertebrae is because of the Hox5 gene. Look in the chick, 1, 2, 3, 4, not many cervical vertebrae. That means they are all expressing Hox5. So Hox5 is telling them to have the cervical vertebrae. But in the mouse, we have only this much cervical vertebrae, then the Hox6 start. Hox6 tell us make the thoracic vertebrae. Making sense? And then you have this Hox9, Hox10, they're again changing the different kind of vertebrae. So depending upon which kind of Hox gene is expressed, that particular type of vertebrae is formed. So the pattern of the body is determined by the type of the Hox gene expression along the anterior posterior axis of the body. So if you knock down these Hox10 from here, mouse, if you knock down Hox10, these lumbar vertebrae will change into thoracic vertebrae. Because then this Hox9 will dominate it. So, what is the take-home message? The take-home message is, we are saying, the anterior posterior patterning of the mammalian development is controlled by the Hox gene. And there are 4 different sets of Hox genes. In each, there are Hox A, Hox B, Hox C, Hox D. In each of the set, you can have 13 genes. You can have 13 different types of, you can have Hox A1, Hox A2, Hox A3, Hox A4, Hox, up to 30. And then we are saying, the type of Hox gene expression pattern changes the type of vertebrae. And we are saying that different expression of different Hox gene control that which type of vertebrae is going to form. And we have given an example here of chick and the mouse. We are saying in the mouse, we are showing here these cervical vertebrae here, and we are saying these vertebrae are patterned because of this HOX5 expression. Here the HOX5, they have more cervical vertebrae chick, so they have more area with the HOX5 expression. Making sense? So they have more. Now we have more thoracic, so we have more area with HOX6 expression. Then the chick, they have less number of this. Making sense? So just tell the expression pattern of Hox can change the expression, not the expression, but the pattern of the vertebrae in the vertebrates. Making sense? Okay. So then this was about the Hox hypothesis. So talk about this thing. We didn't talk at all about even the gastrulation. So talk about gastrulation and talk about this, then we'll go further.
Then they start moving down, and then my understanding is when they meet the visceral cells with endoderms. There's not underneath, because these are endoderms, these are down. Well, so yeah, so I have an epiblast and then visceral endoderm. So they move down, then they make the definitive endoderm instead of visceral endoderm. And then they're no longer called epiblast cells either. They're called the ectoderm. And then the extra cells that can't go into the definitive endoderm become the visceral.
Is there a difference between EP and E-cadherin?
Say it again.
EP and E-cadherin?
Epicadherin or epithelial cadherin and E-cadherin, they are the same. They are the same, yeah.
Moves down to the condition and so into the visceral endoderm layer. And slowly replaces cells. And they keep moving down and they form. Oh, sorry, that is on the two layers. And they keep moving down. After about 16 days, and form the third layer of the mesoderm.
this is just supposed to be like the head is forming in this picture.
When you say the anterior visceral endoderm, is,
I think.
the same thing.
Oh, is this
Like, I think it's just-
It's exactly the same thing, but we don't call
I think
it
it's
new
just-
group
Right,
center.
yeah, it's.
Like.
Yeah, but it's the same thing, it's the
Right.
new group center of the
Okay.
So it's telling
And then we talked about
mesoderm
this.
about to behave
You
like
said they all have seven, and.
the organism.
Change.
Yeah.
But it could be good.
Yeah, so is this only in like humans too? Like, do giraffe have this many too? Well, the Hox genes code whatever, determine the patterns of birth and for the body. And mesoderm gives us. And then all known relevance of forces of body forming which are expressed.
Clear about
perhaps
Hox
in
gene,
concept or
any
chromosomes.
doubt there? Oh.
Their location, order, and expression is similar in all vertebrates. Humans have Hox A, B, C, D.
So when we drew this picture, this is just like the head forming in the spinal cord and then the pylori forming around the spinal cord. right?
Yes, yeah. Yeah, and this Hox gene is about the type of vertebrae formation, right? And the nervous system is more of a BNP and doing signaling. Now, so let's talk about the left-right axis. Left-right axis. Now, in mammals, we know that we have left side heart and all those things are there, right? There is a left-right axis. So, right here you have your, just drawing the cells of the node. So this is your node. Remember that embryo is fluid filled, there's a lot of fluid there, right? So the cells which are here, cell lining here, so these are the node cells. They have cilia. So these are ciliary node cells. And there's fluid in the body. And what they do in the developing embryo, what cilia do, the ciliary node cells, their cilia propels fluid in a clockwise pattern. So in a clockwise pattern. So if you think about from left to right, right? From left to right. So that fluid is flowing. They're doing that. So if people have mutation in diamine, so we say 50% chances heart on the right side. Okay. So why that happen? Why that happen? So the cells, if I draw it like, if I cut this section like this, so I will say, okay, so these are the cells which are the ciliary node cells, right? So they're moving the fluid like this. They have the dynein, they can move the fluid. Next to these cells right there, right here are next to those are crown cells. Now these crown cells, what they have cilia, but those cilia are immobile. They usually don't do anything like moving or like that, but they have cilia. Okay? So what happened, when these ciliary node cells, they are moving the fluid. So right here are the cilia. Right here are the cilia. So they are moving the fluid. And that fluid basically touches these cilia of the crown cell. Okay? So what happened, that the crown cell cilia they sense moving fluid, right? And that sensitivity of the moving fluid, what that does, that activate PKD. PKD is PKD2 in these cells. That activates PKD2 in these cells. And what PKD2 does, the PKD2 activation that activate the expression of nodal and inhibit cerebrus on the left side. Okay? Remember, this node has already determined which is the left side, which is the right side, that the fluid is going on, right there. So when the fluid is moving, so this is the right side, this is the left side. So that fluid moving cause the activation of PKD in the crown cell. And when the crown cells are activated by the PKD activation, what they do, they activate the expression of nodal and inhibit the expression of cerebrus on the left side. And what that does, that cause activation of PITX. Activation of PITX. And when the PITX is activated, that determine activation of formation of left side structures. So what we are saying? We are saying that the nodal cell, they are the ciliary nodal cells, they basically coordinate the movement of the fluids in the developing embryo. And they move fluid in a clockwise direction in a way that the fluid is moving from left to right side, right? And when that happens, the cell adjacent to ciliary nodal cells are the crown cells. They are the cilia, those cilia are immobile. Those cilia now can sense the flow of the liquid. When they sense the flow of the liquid, in those cells, they basically activate the expression of PKD2. When the PKD2 is activated in these cells, that causes the expression of nodal signaling on the left side and the repression of the cerebrus signaling on the left side. When that happens, that causes activation of PITX, and PITX activation determines the activation of the formation of the left side structure. Making sense? So when you don't have that fluid movement, what is going to happen? There is a random chance. It can be activated, it may not be activated. It activates because there are other factors, but where it is going to activate it is random chance. That's why I say 50% chances you can have heart on the right side. Making sense? Okay, talk to each other for a second, then we'll go further.
Random chance that the PIT X is activated or PKD2? You said there's a random chance it's activated, which one, the PKD2?
PKD2 and then PITX, it can happen anywhere,
Okay.
right? So ultimately, you can have then the heart in the other side where it should not be.
So the expression of the nodal inhibition of the cerebrus. Yep, the inhibition of cerebrus and the expression of the nodal.
Yeah, Cerebus is inhibited on the left side. So Cerebus is inhibited on the left side, and nodal is expressed. Activate the expression of nodal and inhibit Cerebus on the left side. But if Cerebus is inactivated, or inhibited, PITX determines the activation and inhibition of the left side structure. This all causes the activation of PITX.
So it's a cascade just to make sure PITX gets,
Your cerebrus inhibition and nodal activation that lead to activation of
together
PITX.
will activate PITX, and then PITX determines formation of left side structures.
Okay.
So if all that doesn't happen, then there's a 50% chance that you have right side heart because it could potentially be activated by outside factors.
Okay.
Okay, now we are on the last thing here, and the last thing is twins. Two type of twins, dizygotic, two eggs, two sperms, right? Different zygotic. So, two eggs and two sperms, right? If that happen, what we call them? We call them fraternal twins. Those are fraternal twins. So, two different fertilization events are occurring at the same time, right? Then, you can have monozygotic twins. So, this has divided into two. We can say that even it can divide up to 8 also, right? You can have 8 twins from one. We don't call them twin, but 8 coming together, right? So, yeah. And then these two can separate very early. Separate very early. If they separate very early, what is going to happen? They will have their own chorion. Right? Because they're separating separately. So 30% of the twin, 33% of the monozygotic twins are like this. Okay. Now what can happen? Say they keep on dividing like this. So the ICM is formed, right? And then the ICM kind of separate in two. So I see them separating two. So what is going to happen? You're going to have a situation like this. A single chorion, one amniotic sac, and twins. 2/3 are like this. Okay? Now, very rare. Amniotic sac formation take place, right? And then somehow they divide. Okay, so even then you can have the two twins. I should have had that here. Let me tell you one thing. When I'm saying they are dividing at this position, you're having two different amniotic sac because they divide. Not one, two amniotic sac. But when they divide like this, very late once the amniotic sac is formed, then you can have only one amniotic sac. So you have the situation, one amniotic sac, and you have two twins. Very rare, very late. Okay, then you can have conjoined twins. And Emily had that answer. She asked me earlier. I told her it out. You can have guest relations starting at two different points. You can have conjoined twins. So what we are saying is that twins are exactly maternal or fraternal. Fraternal, two separate fertilization events, two separate egg, two separate sperm, giving you two genetically different people. You can have monozygotic twin. Technically, genetically they are same, but they are not same. So if someone says they have the same gene expression, that's not true because the epigenetic changes will be very different, can be very different here and here. So if you say that, it means what is the difference between this? They have the same genome from the same parents, right? But genetically they have different. So saying one person is genetically similar to other is a very difficult term to define. When you're talking genetics, are you talking epigenetically similar, or you're talking about the number of genes similar or gene expression similar? So gene expression is different here. Gene expression is different here as well, because it can be different epigenetic changes there. So then, separate pretty early, two separate chorion. Separate little late, you can have two same chorion, but you can have two amniotic sac. And you are saying they are separating too late. You have the two indivisible, but one amniotic sac, one chorion. Making sense? And then the conjoint is the gastrulation occurring at the different points.
So the very rare one is like the embryo dividing into two in the.
Yeah, in the end, the epiblast cells dividing into two. When you have the bilaminar germinal disc is formed, because that is formed after amnion is formed. Making sense? Any question here? Okay.